Welcome back to Biochemistry. Today we are going to know about the Ramachandran plot. So it is also known as the Ramachandran diagram, which was developed in 1963 by Samudram, Narayana Ramachandran and C. Ramakrishnan and V. Shashishekharan. And the Ramachandra plot was in a way to visualize energetically allowed region for the backbone of the dihedral angle like psi against the phi. Psi angle against the phi angle of amino acid residue for determining a protein structure. So basically this Ramachandra plot, it is used for determining the protein structure accurately, and we can check the errors if any error is present in the Ramachandra plot. So there are polypeptide chain. These are the nitrogen and carbon alpha and beta chain. These are present. The carbon alpha chain, that is carbon alpha and carbon bond, they are free to rotate, and these rotation they represent by a torsal angle like phi and psi respectively. So there is a peptide. What is a peptide? It is a short chain of amino acid, and this amino acid in a peptide, they are connected to one another in a sequence by the peptide bond. There is a carboxylic group of one amino acid that condense with the amino group of the another amino acid with an elimination of water molecule. If water molecule is eliminated, so it will form a peptide bond. So what is characteristics of peptide bond? They have a partial double bond characteristic, and these are rigid and planar. The four atoms of the peptide group like carbon, oxygen, nitrogen, hydrogen, they are having in a single plane in such a way that oxygen atom of the carbonyl group and the hydrogen atom of the amino acid, they are trans to each other, not cis, trans to each other. Then what is it? Is a dihedral angle that is defined by 4 points in a space. So in protein, the two torsal angle like phi and psi, they describe the rotation of the polypeptide chain around the two bond on both sides of the C alpha atom, and the bond between the nitrogen carbon alpha is known as the phi bond, whereas the bond between the C alpha and carbon, that is carbon alpha and carbon, it is known as the psi bond. So Ramachandra plot, that is G.N. Ramachandran, use computer model for this small polypeptide to vary the phi and psi to find a stable conformation of protein. So atoms were treated in hard sphere with dimension that were corresponding to the van der Waals radii. So these phi and psi angle, they collide, correspond, and they decide whether to allow or not allow the phi and psi angle conformation of the polypeptide backbone. So we know there are four quadrants that are present like quadrant 1, quadrant 2, quadrant 3 and quadrant 4. So the quadrant 1, this area confirm, we can find left-handed alpha here. In quadrant 2, this is biggest region in the whole graph, and it is used for conformation of the atoms, whereas the quadrant 3, this is bigger than that of the quadrant 2, and we can find the right-handed alpha here. And quadrant 4, it has no frame locale, so this confirm the psi, that is minus 180 to 0 degree, and phi, that is 0 to 180 degree, and is disfavored due to steric conflicts. There is a secondary structure plot, and this secondary structure of peptide, they are a small repeated building block, and they look same because the amino acid building block have similar angle. So we can check that angle on Ramachandra plot. We can tell the different secondary structure, and the plot help us to see the pattern in how the peptide is put together. And the two most common secondary structure, we know that what are they? The alpha helix and the beta sheet. So what is an alpha helix? It is a right-handed helical coiled peptide chain coiled tightly around the central axis, and they are stabilized by the hydrogen bond between the specific amino acid residue. The secondary structure impair the stability of the protein, and it is important for protein folding and functioning. And in Ramchandra plot, the alpha helix is represented by a distinct region that have a specific phi and psi corresponding to the repeated pattern of hydrogen bond in an alpha helix structure. So this region typically falls with a narrow range of phi and psi angle, and it reflects the regular backbone conformation of alpha helix. The beta sheet, if we see, it is a secondary structure where the neighboring polypeptide chain, they are aligned by side and stabilized by the hydrogen bond between the backbone atom of adjacent strands. These are the secondary structure in protein. So these are they represented by specific region that correspond to the characteristics of phi and psi angle that are associated with regular rearrangement of amino acid residue in the beta strand. So if we see, a large chain would impose more contrasting result in limited allowance region in the Ramachandra plot, but their impact is relatively small. So instead, the most significant influence is observed with presence of alpha and beta of the methyl group that is present at the carbon beta sheet, and the glycine with only one hydrogen atom in the side chain has small van der Waals radii compared to those of the other amino acid, that leads to the less restricted conformation space as evidence in the Ramchandra plot. In contrasting, the proline that have a 5-member ring side chain connecting the carbon alpha to backbone of nitrogen that exhibit a limited number of potential combination of psi and phi in its Ramachandran plot. So this residue preside proline, that is known as the pre-proline, and it show a contrasting combination compared to the general case. So the Ramachandra plot is used to help to understand how protein molecule folds and move, and it helps in analyzing of the protein structure. It helps in checking the protein structure by disallowing the region of phi and psi dihedral angles. In understanding the Ramachandra plot helps in protein modeling structure and predicting, which is important in designing a new drug, and it also helps in identifying if any error is present in an experimental or computational protein structure, and it ensures the structural biological research with very accurately. So this plot serves as a fundamental tool for researchers in structural biology, bioinformatics, drug discovery, and it also helps in studying the protein folding dynamics and interaction. So there are some limitations of this Ramchandra plot. These are useful tools for overall structural assessment. However, there are some limits, like this plot simplifies complex protein conformation space in the 2-dimensional representation, and it relies on the average data and may not capture the specific of an individual protein family, and the quality depends on the resolution, with lower resolution potential lead to inaccuracy, and the crystal packaging effect can distort the dihedral angle in the plot, and inadequate representation of the protein families with limited structural data. It allows the region may still include the energetically unfavorable conformation, and it represents a static view, not fully capture dynamic protein behavior. So this overall Ramchandra plot is used to confirm the structure of protein, and it helps in understanding the protein structure by analyzing, allowing and disallowing the region of phi and psi angle. So it helps in the conformation of protein structure, improving the accuracy and identifying the error. Thus, it is important in protein modeling, drug designing, and despite many Ramchandra plot use, it also has some limitation as well, highlighting the need of careful interpretation in structural biology research. Now let's understand the colorful regions of Ramchandran plot. So we have seen the diagram. There is a white area that shows whether the atom of the polypeptide they come close than the sum of their van der Waals radii. These region they are disallowed for all amino acid except glycine is allowed because it is unique and it lack a side chain. Then there is a red region that confirm whether there is no steric clashes, and there are the allowed region, namely the alpha helical and beta sheet conformations. It allow it. The red region, the yellow area we can see here, it allow if there is a short van der Waals radii that is used in the calculation, the atoms are allowed to come little closer together, and this brings out an additional region that correspond to the left-handed alpha helix. So this was all about the colorful diagram of Ramchandran plot. One more thing which I want to tell is that this Ramachandran plot, it is seen some commonly in very proteins but not in all proteins. Many combination of these angles are not seen in all the proteins, and the Phi and Psi angle could be varied from -180 to +180, and it is seen by the X-ray crystallography study, computer modeling, and it revealed the possible combination of angle, and even the NMR it is also used, and the plot of the Phi and Psi angle in the Ramchandra plot is used. So Ramchandra plot shows a statistical distribution of combination of backbone of dihedral angle like Phi and Psi, and it provide an overview of allowed and disallowed region of torsal angle values. So it serve an important indicator for the knowing the quality of the protein and three-dimension structure. So the torsal angle, they are among the most important local structure parameter that control the protein folding. Thanks for watching. Do like, share and